Hello! Today we have a TLC lab for you. And here we have all the things we're going to use. First we have little dropper bottles with all sorts of amino acids, a can of Diet Coke, and some TLC solvent. First thing we're going to do is draw a line right about 1.5 centimeters from the bottom of our plate, and this is where we'll put our spots. We're going to do 2 different plates with 5 spots each, so we try to draw little lines and identify what amino acids we're going to put at each line. We're going to run analysis on 2 TLC plates. First one is a selection of 5 amino acids, and the second one is an analysis of aspartame and seeing if we can see that in Diet Coke. In order to more easily dispense our amino acids, we're going to use a test tube rack, and we're going to just put a few drops from each of these bottles. As we drop the amino acid, just a few drops in each test tube, I have the structure of the amino acid in the lower corner of the screen. The first one is alanine. The second one is lysine. The third one is proline. And then tryptophan. And valine. Moving on to the front row, start with aspartic acid. Phenylalanine, aspartame. This is not an amino acid, but some combination of 2 with an extra methyl group on it. Then in tube number 4 we have hydrolyzed aspartame, and this is aspartame that I've boiled in hydrochloric acid for a while to try to break up the molecule and see if we can identify its individual components. Here's a final look at each of our samples, and our last tube we're going to put some Diet Coke in. Coming up next is a little graphic I made to show what happens when you decompose aspartame. These are all acid-catalyzed bond breakages, and in the process water gets added, and in the first case eliminates methanol, and then creates the dipeptide phenylalanine aspartic acid. Another addition of water through acid catalysis. It's going to break that bond between the 2 amino acids and form aspartic acid and phenylalanine. If I spin the aspartic acid around, that's the structure that we're used to looking at. And now we're ready to spot our TLC plates. First though, looks like we got a couple test tubes out of order. We want to make sure that everything matches what we have labeled on the plate so that we don't get a spot that's in the wrong place. In order to spot the plates, we're going to use these thin capillary tubes. We just call them TLC Spotters. They pull up just enough liquid in there via capillary action, and then they're thin enough they can make just a tiny little spot on these plates. So we're going to put one in each tube, and it looks like I got just a couple more out of order. Last minute switch here. In order to spot the plate, we just very quickly touch it, and you'll see that one dot dispenses from the capillary tube. We're going to do that with each sample. So we started with alanine, lysine, now we go to proline. And tryptophan. And valine. We'll come back and spot them later and want them to dry first. On our next slide we do some aspartame, followed by aspartic acid and phenylalanine. Then we have our hydrolyzed aspartame solution and a Diet Coke sample. Once they're all dry, we can come back and spot them again several times in order to increase the concentration. So this is obviously sped up. In our lab procedure it says do every spot 4 times and do the Diet Coke 20 times. That's because the concentration of aspartame is less in the Diet Coke. Once we have our plates spotted, we can make our TLC solution beakers, or development chambers, some people call them. We're going to put a little TLC solution or solvent mixture in each beaker, maybe about 25 milliliters. We want to make sure that that solvent is going to be below the line in our plate, which is why we drew the line at 1.5 centimeters. We're going to add a little piece of filter paper to each beaker. This helps the vapor environment in the beaker be a little more even, and then our plate will develop more quickly and have less chance of drying out. And here we're going to do our last Diet Coke spots in order to get up to the number 20. Once we're ready to put the plates in the chamber, we'll take the watch glasses off the top and then just very carefully set it in. We don't want it touching the sides of anything. Get that lid back on, and then here you can see the solvent just creeping up the plate. And it only takes a few seconds to get up to that line where our spots are. After that, it's going to begin carrying them up the plate. This is all shown at regular film speed so that you can see how fast it goes at the beginning. Here we come back a few minutes later, and again later. This is about a half hour. And then after almost an hour, we're fully done, or we're tired of waiting. We're going to take the plate out, and as quickly as we can, without dripping all over the place, draw a line where the solvent front has gone to. Before it evaporates, we need to get this line written across. Now that those lines are drawn, we can dry off the solvent. So here I put it in the fume hood, and we're just going to gently heat it with a heat gun, get that solvent to evaporate. As you can see, initially there are no spots, so we spray it with a ninhydrin stain solution, and then this ninhydrin is going to react differently with each of the amino acids to make a different colored spot. We can develop these spots by continually gently heating with a heat gun. It also works to leave it under a hot light bulb or something like that, just this is way quicker. Once we get those all dry, I can grab a picture, and I put them on the copy machine and got a scan for you too. To analyze the TLC plate, the first thing we're going to look at is called retention factor. Each of the spots on our plate travels a different distance depending on the chemical structure of these different amino acids. It also changes the color. What we're going to look at is the baseline, the distance of the spot, and the distance of the solvent front. The distance the spot traveled is D1, the solvent front is D2, and the retention factor is just the ratio, D1 to D2. This is important because if you had a smaller TLC plate, you'd have a smaller D1 and a smaller T2, but the RF should be the same. Here's another look at both plates, and we're going to look at the aspartame plate this time. On that plate we have phenylalanine and aspartic acid, both just plain amino acids. We may see some of the phenylalanine aspartic acid dipeptide. We have aspartame, the full molecule, and of course Diet Coke on the end. So here's a look at the plate. You can see the aspartame makes an orange dot. The aspartic acid makes a purple dot that didn't travel very far. Phenylalanine makes a dark purple dot. And in the hydrolyzed aspartame column, you can see that we have little bits of each. Now we went over all that pretty quickly, so if you need to go back and review any of these parts, just look at that part of the video. Thanks and have a great day.